So I'd like to welcome everybody to our second seminar as part of our potato breeding and genetics SolCAP workshop. And standing up here, I feel like I'm a cross between a NASA scientist and also some musical stage act. I don't really know how to describe it. So, but our next speaker today is Dr. Alan Van Deynze from UC Davis. He's on the faculty, plant science faculty on campus there, and he's on our SolCAP project that is leading the SNP marker development and analysis at UC Davis.
Okay, thanks, Dave. The handout people are giving me clock already, but I only have one page and it's an outline. And that's because I took the word hands-on workshop here literally, and we're really, everybody should be able to participate. That's on at the class. What I'm going to
for people that are on the webinar, they won't be able to participate unless they have access to that software, which is proprietary. Yeah, so you have to have a license for that software. And we're very fortunate that Illumina was able to give us a license to have this class and able to download on all these computers and stuff. So it is proprietary software you do have to download to be able to use. Yeah, there's a lot of background, so you can maybe turn your... So if everybody can click on the Genome Studio icon on the, they should be at the top left of your computer, we'll get started.
Speakers down to 0.
Is that better? Oh yeah, no backup. So if everybody can click on Genome Studio, the GS, hit cancel on your first screen because we don't need to register, and then the software should mount up right away.
several people don't have that icon.
Is that right? GS. If you don't have that icon, the shortcut, if you can go into Programs, it should be under Illumina. Click on that, and then you should see Genome Studio there. Does that work for everybody? No, you don't skip the username program. So I'll do that again. So for those that don't have the icon, go to Programs, Illumina, click on that, click Genome Studio. Is that right? We're going to have to share then. And that's odd because we thought we had all the computers. So we apologize for that. We thought we had all the computers. So we're going to cancel out of the information screen, and then the software should open up from there. So when you open up, what opens up in Genome Studio is a series of windows, and the first thing we're going to do is actually load a project. And to do that, we're going to hit File, like you would always. You would typically start a new project. We're going to go to New Project, and then we're going to hit, it's a genotyping project. Genome Studio has also sequencing modules and things, which we're not going to cover today, but should have genotyping. Project should come up, and then the wizard basically opens up and starts to ask you a couple questions. And what we're doing is basically going to load the data to tell the software what project we're going to work on, what data we're going to work on. So you click on the wizard, and the first thing is ask you, says where you're going to save the data, the project repository. And so we're just going to hit Browse. And in this case, you could call, if you go under SCF Sol, under My Documents, you should see one potato test data. We're just going to click that. and we're going to save on the folder called One Potato Test Data. So that's where we're going to save our project to. So has everybody got to that? That has the software? So we click OK, and that fills in the window. And the next thing is you have to name your project, and you can call it whatever you like. I'm going to call it Project 1. Put in the name of your project, and you click Next. And then when you click, so you do this, so every time you get new, basically a new set of SNPs, you need to do this, or a new project that you might separate out. Is there a question? Oh, okay. You just said a new person. Once you hit that in the wizard, it's going to ask you, do you want to load from a central directory of files where your genome center might load project data, or just have some local sample sheets which you have. We're going to leave the use sample sheet to load sample intensities. We're going to use that, which is the default. I'm going to click Next. And then it asks basically 3 things that it needs. And so you're telling the software where your data is going to be. First thing you want to know is where your sample sheet is. And you should see this screen. You should go to the One Potato Test Data. I hit Browse, sorry. We hit Browse on the first screen. And it's asking the question, where's your sample sheet? Which sample sheet do you have? In this, so you get a separate, every time you have a new set of samples, there's an associated sample sheet that basically your lab is going to generate to get the data. So they'll give you a sample sheet. This one is called Potato Sample GS. So we're going to click, and they're just Excel sheets with information. And we're going to open that. These are comma separated files, as they're CSV files that are automatically generated by Illumina when it collects the data off the scanner. And basically, so your lab is going to give you these. So that's the first. That's the first line telling you where's the sample sheet. Then it asks the question, where's the data? So you click Browse. Let me double check. We can click Browse. If it goes back down, no, it doesn't remember. If you click Browse, we're going to have to find this potato test data. So SCF Sol, My Documents. And in this case, instead of pressing One Potato Test Data, we go right to Data. And then we'll just leave it at Data. I'm sorry, we're going to hit Potato A1 in this case. That'll be our first set of data, Potato A1. So did everybody get to that? So actually the address to where it should be on your computer is there as well. Documents, Potato Test, Data, Data Potato A1. So what that is the actual data that the Illumina collected, or your lab collected. And finally, there's a manifest. You figure out where is that going to go. We're going to go back to Potato Test Data. So Documents, Potato Test Data. And then we're going to hit Next. And then you get this file, and it asks a couple questions on what do I want to do when I load the data. We want to cluster the SNPs automatically. You can do this later or right now. I just do it right now just because it becomes automatic. So the program can auto-cluster SNPs, and I'll explain what that means once we have it done. The repository was just potato test data, the parent folder. It's the same as the sample data sheet folder. We're going to calculate the statistics around the SNPs. Right now, we don't have heritability data associated, but if you have, say, a population where you know there should be it could be a diversity panel where you already know the pedigree of different accessions in your data, and you have that data in a separate file, you would calculate that. So we're not going to do that because we don't have it. Gen call threshold. Gen is basically a Genome Studio. They have a threshold basically that they calculate when they look at SNPs, and it's something that's calculated in the background. For Infinium data, this should be 0.15, and that's the default. For Golden Gate data, so these are usually samples less than 3,000 SNPs, that should be set to 0.25. And so this is just an internal call that they use. But right now, let's just leave it as a default 0.15 for the Infinium data. We'll just hit Finish. And then it's basically, now it's loading all your data. And there's a number of windows that open if everything works out. And there's actually 5, 6 windows that open, and there's a number of them that are a waste of time, that are basically taking up space. This is just a log on the bottom window. So I close the bottom window. So you notice it just increases the size of your window to full screen. The errors table we're not going to use. Let's just drop that. And the window that we like to see a lot is actually this one up here in the top left. And so we can actually make these windows as big or as small as we like. So something like this is a good way to work. If you want to see what windows are available, you can actually go down to the windows and click. You can click and unclick different windows as you want. You notice there's 3 main windows, but there's more than 3 windows clicked here. But notice there's some tabs in these other windows. So these represent these tabs in the window, like the paired sample and the SNP table, which we're going to go to. So one of the first things that you want to do is really define the clusters. And what a cluster is if you just click on any one of this. Let's click on number 1, the top window over here. That's a bad example. Let's click on number 2. And what the software does, it color codes the clusters, basically where it thinks the genotype classes are. The clusters that are in the red are what's called the AA genotype. And the A genotype is actually the genotype, the first genotype that we gave it. When we define the SNP, we give them a sequence and we say this is a CG SNP. The A is the C, the B is a G. But you don't have to remember that because it's all in these tables, which I'll show. Okay, so the question is, so what kind of data are we looking at? The data is this particular data that we're talking about is a set of 96 SNPs that we developed with the SolCAP project to validate kind of how good our SNP detection algorithms are. And so you'll get this. What's unique about all data sets is basically how the SNPs are defined. That's the first thing you need to know. And that's called your OPA file, which was one of the files that you're going to find that was already loaded on the computer. So you need what's called the OPA file. And the OPA file defines what sequence the SNP what the SNP is and at what position. And it basically has the criteria of what the sequence, what the SNP is, what the allele is. Is it a CG SNP, an AT SNP, and so on. So, and then the names of those SNPs. The names of the SNPs in this case are in the table over here. If I stretch this out, name of the first SNP in the table is SolCAP C2 13199. So that data is captured in the OPA file. These are predefined SNPs, exactly. So this is the Illumina genotyping. It's a true genotyping platform. It's not a sequence platform. Okay, so these are SNPs that are predefined. You've designed an Illumina assay on, and we're not going to talk about how that's done. But so SolCAP in this case, we worked with Robin, who designed SNPs based on the alignments that she was talking about, and some other data that we generated. And we send the sequences to Illumina, and they design an assay around it, basically a set of primers that they send to. They design basically the physical oligos for those primers. They put on an Illumina chip, and what the Illumina chip actually looks like is an array, it's like a glass slide, and in one well, so to speak, or one pin. In this case, there's 96 SNPs. So you add one sample of DNA, it'll interrogate 96 SNPs. What you're going to see in the Infinium array that SolCAP is generating is we're going to have, there's actually 10,000 data points on this array that we're generating, and it will represent about 8,300 different loci that we're interrogating. But it's still the same thing, one sample, 8,300 loci. In this case, there's 96 lines. If you scroll around to the bottom here, there's 96 different samples or SNPs that have been interrogated here. And what we're seeing is the data generated from that. Well, in this case, there's SNPs within genes, but they don't need to be. It's whatever you've submitted to Illumina to generate. It could be any SNP that you want. in SolCAP, they're all SNPs that are from express sequences. So I'm saying they're express sequences. They may not be in genes. They could be 3' and or, you know, and so on. But they're part of, yes, so they are in genes, not necessarily expressed. I'm not positive on actually our nomenclature on that. I'm going to have to ask Robin. The C2. Right. So,
the first one will provide you the actual transcript sequence that that came from. Will provide the location of the SNP and what the two alleles are. And as soon as the public genome sequencing presentation is completely available, we'll be able to put that right on the genome browser. Like what polymorphism is. Right now we can't. No, but you could take that config and you could blast it. And you could easily find out. That was fine. I feel that good enough time. This time you like to put that. Oh no. That data I think just on the website just
okay. So the question was, what do the numbers mean or in the name? And the answer is not very much. And there's going to, as far as you're concerned right here, but we have a cross-reference table at SolCAP, which we'll put on the online shortly, which will show what the sequence is, what the SNP is, and so on. But I'm going to show you, there is, in the software, it'll tell you what the SNP is, actually, in these tables. You can capture that. But the actual sequence that it's from and so on, that'll be something we'll cross-reference as part of the SolCAP project independently. That's what I tried to say. I just did. So, yeah. So basically, this software is not fully integrated into the genome browser or any of the other SolCAP tools. This is independent software. And what we've loaded basically is a set of SNP. We told it what the SNPs are. We told it what the samples are, and the software is capturing now, putting together what the combination of those two are when we put them in the lab. We put them through the lab. And what you see is really graphically in the top left corner is the results of putting basically the SNPs and the samples. And what you're seeing is all the samples at once. And it automatically will try to cluster the data based on what it sees. And that's going to walk through and how, but you don't have to accept their algorithm. You can refine, you know, anything you like and make these clusters tighter, or say, you know, I really know that the heterozygous classes should be out here somewhere. So I just clicked on a marker here, the number 5 in this case. But to change the clusters and what to keep, you can hit the Shift key. So you press your Shift key, and you can say, and you see this crosshair when you go over the middle of it, and you can say, I really think the heterozygous cluster should be a little bit over to this side. So what did that do? So all I did is I shifted to the right. Notice when I do that, the purple cluster, now this point down here, the point in the red that is now red, and that's off the axis, is now going to be called an AA allele instead of an AB allele, a heterozygote, because I've moved that cluster. You can do the same thing here to say, you know what, I'm not very confident with these guys on the edge. I don't even want to make that call. So you can actually, hitting the Shift key, you see these, you can make this a little tighter, and you notice it's going, and you know, and now I'm excluding this data point. So I'm actually going to make the red a little tighter, so I see the kind of the size window. I'm going to make this really tight, and notice this guy, which I'm not sure of, it turned black. That's going to be a no call in your data now. So you can kind of exclude data points and say, I'm not sure. I really don't want to make those calls.
Since these are all tetraploids, should there be more genotypic classes?
We're gonna go there. So the question was, since these are tetraploids, should there be more genotypic classes? The answer is yes. So this software right now is only set up for diploids. So it's only going to call headers, 3 classes, homozygotes and 1 heterozygous class. And we're working actually with Illumina to add that feature, but we've found a way right now to do it, and I'll show you how to do that independently. using some Excel macros that we've developed. So right now, just assume you're working with the diploid because that's all we can call at this point using the current Illumina software. But within a few months, they hope to have Polyploid, a module for this. We provided them actually with this very data set, and they're going to work with that to make those calls. Pardon? Oh, what are the graphs? The graphs are basically a combination of intensity. Let's make sure I get this right. The graphs on this scale is basically the intensities of red and green. And theta is a measure of that. And that's really what you're looking for on a scale of 1 to 0. So where the A's would be all red, and then they actually capture green, and these look more yellow. So this is kind of the fraction of green and red mix is what you're capturing in this. So we're defining clusters while we're doing this, and you can do this by you have to hit the shift key and see. Notice that the circle went out kind of into this homozygous class over here. You can shrink that and make it as tight as you like. And see, but notice that I've made the circle a little tighter. I've also excluded these data points. Now these data points are going to be excluded, are basically going to get a missing data point. Anything black is a missing data point. It's not going to make a call on it. The other thing I want to show is you can only change things in what's called the polar. This is called the polar view. But if you click here on the second little graph, it's called the Cartesian view. And this is looking at the relative intensity using a little different algorithm. And I like to look at it. You really see the clusters a lot better in this view. I'll try to pick a SNP. So for example, if you look at SNP number 4, you can start seeing that there's clearly 4 clusters there in this tetraploid data. And you can count, there's 5 clusters if you click on number 6, for example. It's not perfect, but when you look at this data, you can actually start to see it says 1, 2, 3, 4, sorry, 1, 2, 3, 4, 5 clusters there. And we're seeing, just to give a little background, we have 96 SNPs here that we're interrogating, and we're able to call 5 clusters using our software, which I'll show you, in 56% of the data points. So 55 data points, we actually saw 5 clusters that we could make sense of. And we saw 4 clusters in about another 25% of those data points. Just to give an idea of the sensitivity of the Illumina assays, at least for this set of data points. So for over half of the data points, we're able to see basically the 5 genotypes that we expect. So does everybody capture the 5 genotypes? So it's basically with 4 genotypes, first is 4 A's all the way to 4 B's with different doses of A in it, and you get 5 possible genotypes in a tetraploid. But for now, let's just work with the diploid features, and then I'll extrapolate this, and then we'll show how the tetraploid data can be analyzed. So you can basically capture different. You scroll through the different markers, and you start seeing. So here's a nice one, number 20, where you see the 5 clusters pretty nicely. And to get the different, but unfortunately you can't change any of the clusters here. To change, you have to go back to the polar view, and you have to start playing with using the shift key and changing where the clusters are. What you really like to see in the perfect data point is all the A's. Basically, you want to see a cluster on each right tight to the axes. You see, you got homozygotes on the y-axis. You got homozygotes on the x-axis. That's a nice clustering. That's a marker you're really confident in, that those homozygote classes are really tight. And in this case, you could really see, I think, the 5 classes quite nicely. Let's see. Does Illumina make a mistake? Absolutely. Well, Illumina has the same, is limited to the same problems we all are. It's garbage in, garbage out. If you have good markers, good primers, you're going to get good data. If you have good DNA, if your DNA samples are not great, if they're not normalized to a fairly uniform concentration and so on, that's going to play into effect on how good your data is going to be. Actually, number 9 is not that bad. I'll give you an example. The best way to figure out good from bad, and this is kind of the next thing. If you click on GenTrain score under the full table view, you now have sort tools. Why don't we sort from top to bottom? The GenTrain score is the confidence it has in calling all the data for that particular SNP in this case. All right, so he says, so click this guy. So the one at the top has a really low GenTrain score, and notice that there's no homozygotes there on the x-axis. Everything's kind of bunched up really tightly, so it's not very confident in those calls. So that's a good way of actually, instead of going through 96 samples, you can say, well, I'm just going to look at the ones with the bad gen change score. And what I found, though, is you really have to look at both ends, the top and the bottom. So if you click anywhere in the middle of GenTrain, these are nice, these are beautiful clusters. Those with kind of 0.5 GenTrain scores, those are really good. But the ones with really high GenTrain scores are really bad too, because it's really confident that that marker is not polymorphic. So it has a high GenTrain score, but it's a really bad marker because there's no polymorphism in it. It's all one cluster. So you got to look at both sides, both ends of the data. So that actually, this is the perfect timing on the question. I mean, it's a great way of looking right away at which markers are good, which ones are bad. Can this technology handle more than 2 alleles? Well, the answer to our data the answer is yes. Look at the GenTrain scores in the middle. You're seeing there are 5 clusters. Can the software handle it? The current version, the answer is no. So the question is, can this technology handle, say, more than a SNP that has more than 2 alleles? The answer is no. But is that something you want to do? Probably not. It makes it pretty difficult, actually. And for most part, we haven't seen too many of those in our data. We've designed only what we've seen, at least on the current sequence, biallelic SNPs, as opposed to triallelic. And you rarely see, you see triallelic, but they're not that common. So the question is, if you're testing a wild species for a particular SNP, are you likely to detect this? The answer is we do actually have quite a few species here. We did actually do this data set with a bunch of diploid species and so on like that, and we did get good data from that. So that's kind of the same question that we had in the previous seminar. If I design a set of primers in the DM, can I get it in in the others, and so on? And the answer was yes. For what we've seen, the answer is yes. You may get some that fall out if they're too broad, but that'll show up as missing data points. So from what we've seen, we're fairly new on this. We've tested about a dozen diploid species and about 500 lines in total, actually, with this data, and we've had very good success rate on the calls. We had very few missing data points. So the answer is, the question is the number of SNPs quite limited across species? I don't know about the number, but the actual position is conserved. The SNPs are conserved. As long as the sequence aligns and it's not an insertion deletion, there has to be an allele at that place, right? Is it a new allele or not? If it's actually a C where we've only interrogated an A and a T in the SNP, the assay won't work, because the assay was designed for an A and a T. It wasn't trying to interrogate a C. So if you actually have a C in your wild species, that space, it'll be a missing data point. So it's limited to the assay. If you have a CG SNP that you're assaying, that's all it's going to assay. If that sequence doesn't exist in your wild species, it's obviously not going to show up on either the C or the G SNP. But from what we've seen, the SNPs that we've checked, it's been pretty useful across the diploid species that we've tested, and for the vast majority of the markers, actually. So I'm just going to kind of go through the different options that we have here. You can actually, what's nice about this software, you can click, and when you right click, you can get context specific menus. And one of the things you can do is, let's see, what did I want to show here? You can choose different colors for different. Notice here I have one sample highlighted, and the sample that's highlighted is in yellow in this case. I can actually right click and say configure mark, and I can add, and I'm going to say I'm going to call this data point point 1. You know, you can call it a set, and I'm going to, then you can choose what color those points are going to be. I'm going to say everything, I'm going to call that data point, I'm going to show it to be, it's going to show as white from now on. I hit OK, I can hit OK, and now that data point, no matter what SNP I have, is going to show up white. It shows up white. So you can highlight some samples. They could be your control samples and so on, things like that. You can do this in, you can highlight a number of data points. If you say, well, I really, it's called lasso mode. You can click this little lasso at the top. You click that lasso mode and you can highlight a whole bunch of data points in this area. And now those are highlighted as yellow, and you can define those as separate things. So think about this as, let's say, in the context of clusters, if you see different clusters that are not highlighted here, you can highlight the clusters differently. But those clusters, right now I'm highlighting samples, not highlighting SNPs. So those clusters will change with the different doses of those SNPs, of course, for different SNPs. What it does, so it also highlights in the sample which samples I've highlighted. Is there a reset key? Just click out of it. Just click out of it, and then the highlight's different. So the yellow guy. So the yellow guy to reset, I think you have to just, you'd have to re-click. Forget if there's a reset key there. Oh yeah, there's a clear. That's it. Yeah, if you right-click, there's a clear marks on. Clear marks. That's it. You can all or clear the point 1. You can decide which cluster you want to clear, and then I can just hit all. Let's see. One of the things that you can look at, if you don't like, so they've chosen red, purple, and blue, which if you're colorblind, you might not see the red so well. You can choose what colors you want. So if you go to options for the project, you can actually choose what colors you want for the different backgrounds that you like. So the A is always red in this case, purple for the heterozygote, blue for the other homozygote. You can exclude samples. You can actually highlight samples to say I don't want them anymore, and those can be highlighted in different ways. So if I highlight a couple, how do you exclude a sample? So you say this is a bad sample, I don't want to see it. You click on the sample, and you can just click exclude selected sample. And by doing that, basically the data will not be collected or presented for that data point. And you can do the same for SNPs by clicking on a SNP. I guess not in this set. I'll show you in a report. You can actually export only certain SNPs if you want. One of the, if you forgot to click, if you want to recluster, if you don't like to see the shades, you can hit this little shade thing, and you can take the shading out. You can also move this up. You can also move this up and down. You can zoom in to certain clusters if you want to be able to fine-tune these a little more. And so on. Okay, I want to zoom out. And to get back, you can just hit these guys. So this optimizes your screen horizontally, this optimizes it vertically. If you want bigger, smaller dots, you can do smaller dots, bigger dots. Let's see, that's the first part. If you want to auto-cluster all the SNPs, this little guy here, when we started our project, if you forgot to click auto-cluster the SNPs, we can just hit that. And it'll, so right now, if I auto-cluster all the SNPs, all the movements I made before now won't be saved. So I hit yes, it redoes, basically it goes through and calculates the statistics again. So you can do that at any time. So now, so we've moved around, we've defined clusters. So now we want to be able to export data. So if you want to export data, or, I'm sorry, create. Let's look at the SNP table. So if we click at the SNP table, you have a whole bunch of different things that you see at the SNP table. You got your name. We never defined the chromosome because we don't know it. But you see chi-square tests for basically for a 1:2 to 1 in this case. And there's a number of statistics here. So you have the frequency of A's and B's, the call frequency. So this is something you might want to sort by for your SNPs. So let's sort by call frequency. We got 100%. You know, let's see some of the guys in this data set. Basically, this data set has a very high call frequency. So there's only one SNP that really failed, that had poor calls, and had 0.56.
Call frequency, that means that in your reference clone, it called it 100% of the reference clone. Is that correct?
But no, not to reference all your data. It's looking at all the data that you've loaded here. So in this case, there's 96 samples. It's called only half of those samples in this SNP that I did, 56% of those SNPs. So the call frequency is very high. So you notice that there's one at 56%. The rest are 90 and above. So it's able to make a call. But you can change that by playing with those clusters like we did. You know, you can exclude or include samples. That's not the allele frequency. So that the call frequency and the chi-square are not directly related, because you may have called only 50% of that data, but that data might still be in equilibrium 1:2 to 1. But you know, you only have 50 samples now instead of 96. But there's 12, 25, and 12. You know, to give you a high chi-square in this case. So they're not related at all, actually. Minor allele frequency, that's basically the B allele. These 10% GC, 50% GC. GC is gen train call. That's just a percentile. I mean, it's the percentile of SNPs that you get a gen train call score, and this is basically the score that's the 50 percentile, the 10 percentile. So this one has a high gen train score. The 50th percentile is 0.28. Those are relevant. But what you do want to see is, in this column, the SNPs. So it tells you what is it. It's an AG SNP in my case that I'm showing. 95 of the samples out of 96 were actually called, and there's only one missing. Let me see a number. So I'm not interested in all these columns when I want to export things. I can hit what's called the column chooser, and that's this little button over here on the top right. So it's this little, does everybody see that, the column chooser? So the column chooser is a little button over here, and you can decide which columns I really want to see. So some of these are hidden. So in the display column, so I want to see the name, the chromosome, the chi-square. Let's say I'm really not interested in this auxiliary column and everything in between. So all I've done is I've hit the shift key, hit everything in between, or I hit the control key and highlight things or unhighlight things, and I hide those. This is the name of my OPA file. I don't need to see that, and so on. So I hit OK. It reconfigures it, and now it's only showing the columns that I've highlighted. And so that's important. Actually, I'm going to hit choose cooler. There's nothing in the chromosome position or the position. I'm going to hide those guys. So now I've got the index, the name of the marker, the chi-square, and basically the calls, the allele frequency, and the SNPs. So I can actually, so to see what I want. If I go back to full table, that's really your data. You can do the same thing. So you choose. I don't want the chromosome. I don't want the position. I really only want to see the genotypes. So let's see. So the calls. So notice you're starting to see your samples. I'm going to highlight everything between here, hide the rest, and this will be the data for your samples. So these are the name of the samples that are in there. So I want to see the data for all these guys. I'm going to hit OK, and then I can export that data to a file. I can just export. There's just a little icon over here, export display data, and say where do I want to put that? And so it automatically will go to your project file, give it a name, call it full data table. I'm going to call it 1. I hit save. Do you want to only? So this is a chance. Can I select just certain SNPs, or do I want to export everything? I'm going to hit yes. Do I want to view the file? I'm going to say no. So I've exported that file right now, and you can upload that into Excel and then upload that into something until your favorite software after that. And you can do exactly the same thing on the sample data sheet. You can choose what you want. These P are really percent. So the 5% green, 50% green, these are like medians or percentiles on the green-red scale. They're not, to me, they're not that informative to me on the sample sheet. So if you want a sample sheet, you might just highlight everything. And it just shows you that. And to undo that, you can just, if you want to see everything, you can just highlight and then show the column you like after that, and so on. So what I'm going to show you now is something SolCAP is going to do and provide for basically our chip arrays is what's called a cluster file. So we're going to go through all our SNPs and we're going to define the clusters. And instead of everybody having different cluster files and not having the same calls necessarily, you can export that data, and then you can import that cluster file from SolCAP and say, okay, this is the way SolCAP would have called it, based on the data set they have. The question is, how many SNPs do we have? Right now, we've only collected data on 96 SNPs, times about 500 samples. Yes, we don't know where they are in the genome. The SNPs were chosen, of the final array, the SNPs were chosen to basically represent as many scaffolds as possible based on the genome sequence that Robin talked about. And they were also chosen on candidate genes that people were interested in around the carbohydrate pathway and then some others. So that's the general selection of SNPs. And the criteria for that is actually is on the SolCAP website on how we selected those SNPs. So we haven't mapped these. We don't know where they map exactly, but we know what sequences they are and where they are, so which scaffolds are associated with on the genome, essentially. So that's what we know for now for this. So these are SNPs that occur between the GM and the RA? No, these are SNPs. Most of the SNPs were between the 3 varieties that we sequenced: Atlantic, Snowden, and Premier. And we did the transcriptome sequence only with Illumina. So they represent SNPs around genes. Would that be accurate, Robin?
Yeah, so I mean, to be honest, yes, I know where all the SNPs are, but you guys can't know probably like 2 more months. So we're just now preparing the annotation file to go with the SNPs on the array, and with that we'll put, here's the sequence they came from, or the transcribed sequences, and it's either from Atlantic, Snowden, Russell tell you which scaffold you're on in the double monoploid assembly. We'll tell you what gene it encodes, and we'll tell you exactly where the SNP is in that sequence. And then hopefully in like 2 months, the Potato Consortium will release the pseudomolecules, that is the 12 single sequences that represent the 12 chromosomes, and then we'll tell you where on those.
Okay. Okay. So the answer was how do we know where the SNPs are from? And the answer is, so right now the public can't know that. In 2 months, Robin will put that. Robin knows where they are in the context of which scaffold they're on, what the SNP is, what the base of that SNP is, from what variety. We used the 3 varieties, Atlantic, Snowden, and Premier, but we also used the EST sequence and the genomic sequence that was available in the public as well. So there was old variety EST databases from Canonbeck, and then also the RH and then the DM. And so stay tuned for about 2 months, and you'll have really the kind of the whole history and be able to follow where that SNP is really from. And we'll have all that data on the SolCAP website link. So the question is, can you link phenotype data in this software? Generally, so this is not QTL analysis software in any way. This is just to manage your SNP and export genotypes for the most part. Oh, as far as annotation, you mean? This software, actually you can annotate SNPs within this software to do that, but I don't think this would be the best spot to do it, to be honest. You're better off to get your genotypes and I think probably use something more integrated as the tools develop on that. They do have a phenotype column that you can add phenotypic data in CSV format, but all it really does is really display data. They'll show your phenotype red and your SNP red, so you could kind of make pattern type associations in a heat map, but there's no statistics around it whatsoever. And so it might be pleasing, but I don't think it's going to hold water, and I wouldn't be making any decisions on it, to be honest. So the answer is you probably shouldn't use this really as a phenotype genotype tool. Okay. I'm running a little short on time, and what I'm going to show you a couple more things is I'm going to pick a SNP that's fairly clean. So I sorted back by index. So I just hit the sort key. I went over index, hit the sort, went index, sorted, and I picked SNP number 4. And what I'm going to do is I'm going to shift. So I'm going to shift a couple things, and it doesn't matter what you do. I'm going to shrink this, and I'm going to move this. And so now I've changed from the automatic clustering file. What I'm going to do is I'm going to export this cluster position. So this is what, say, SolCAP will do. We'll define all the clusters, and we're going to export cluster positions. So a cluster is basically these different shades. This is a cluster. The purple's a cluster. Clusters of SNPs. This is a heterozygous cluster, a homozygous cluster, another homozygous cluster. Exact cluster. It's really clustering of genotypes is what it is. So you can export basically all the clustering, basically all the defined clusters or the data for this. To click File, I export the cluster positions, and it's say for the selected SNPs or for all SNPs. I'm going to hit for all SNPs. So File, Export Cluster Positions for all SNPs, and it's say where do I want to put these files. It goes right back to that directory we defined initially. Let's call it cluster 1. And we hit save. Okay, so that changes nothing. All we've done is export the current data. I'm going to hit recluster my files back to the automatic. So I'm going to click this cluster all SNPs again. So that the little 3 marks over here, hit yes, calculate the data, yep. Notice it went back to the original thing. Okay, so we're back to auto. But that's not what we want to see. We want something that from this, I'll give you an example. You know, I want to do what SolCAP did. I go back to file. SolCAP will have, you know, on our website, we'll have, say, a cluster file for these SNPs. We're going to import that cluster file. And then we see, so we're going to import the EGT file. That's a cluster file. And in this case, it's the one we just did. So we hit import, choose our file. We're going to open it. So now it reclustered the files the way we had it reclustered. You know, so it's going to put the data in the context of the predefined clusters that somebody else did in that cluster file. So that's actually a very important thing to know, so that you don't, for example, we'll have 8,300 markers in our Infinium array. You don't want to go through 8,300 markers. You know, let's one person do it, and then everybody use that data set. So that's something really important to know. Well, it won't be in the help file, but it'll be in the SolCAP file, exactly. You know, but of course, you have the option of saying, you know, I didn't really like what SolCAP did. You can move those around later yourself. I mean, your data may look different than ours as well. So we've covered, you know, we've played around with the clusters. We've selected low-sign SNPs. We've sorted samples. We've exported clusters. What we're going to do now is going to add new data. So this is 96 samples with 96 SNPs. I've run a new plate. Let's combine two data sets with these 96 samples with 96 new samples. So you can go File, Load Additional Samples. And we're going to, you see this screen, which you've seen before. Use sample sheet local intensities. We'll click the default, Next. It needs to know what those samples are, so now you need a new sample data sheet. We're going to go Browse. And the second sample sheet, in this case, is called Potato Control Samples. So we're going to click that, and we hit Open. And where's the data? We're going to go back to my Documents, Data, and we'll call the, you know, we'll capture the data for that plate as well. So click SolCAP Potato Control. Okay. So that's the two things it needs. What are the samples? Where's the data? It already knows where what the SNPs are because that's in, it's the same SNPs as the previous set of samples that we analyzed. We click that. And then if you look on the bottom left, now it's displaying 192 rows. So there used to be 90 times 96 SNPs. So there's 96 SNPs, but 192 samples on that. So you now have new samples. Some of these samples, let's see. This control plate, what we've done with the control plate is we've actually spiked some controls in to try to define the cluster. So we spiked it, we used a double monoploid, and we spiked other diploids in there in ratios of 4 to 0 to 0 to 4. So 3 to 1, 2 to 2, 1 to 3. And so we should be able to see where those samples are if they're polymorphic, if it follows the dose. So I'm still on marker number 4. And let's look in the Cartesian view. It's easier to see. And if we click these, you can see these samples 3 to 1, 1 to 1. And you notice the dose is moving. Or actually just highlight the samples. Number 4 SNP. Let's highlight samples 102. Scroll down to 102 to 105. And this is what we get. We see a nice dose response in the clusters. So this is a control plate that SolCAP put together. We capture about 40% of the loci with basically with about 3-4 mixtures of diploids. So this is what we're going to use to help define those clusters. We have spiked samples that we see the dose. And we're seeing this works pretty well. So you can clip through markers. So this isn't going to work for all markers. So here in this case, DM times XO8 is not polymorphic when you click the marker number 13. So it doesn't work for all the markers. But we have, and you'll see the doses here, so we can clip the different markers. So if you highlight 109 to 113, you'll get different doses. This one doesn't seem to separate out as well as the other one. But you can walk through and you can see, you start defining doses and so on with spike. So the question is, I expected to see the points in the center of the cluster. That's a comment. Then it's not perfect. I mean, the cluster, as you can tell, if it was all perfect, you'd only see 5 data points, 5 points there with a bunch of on top of each other, right? I mean, there's movement due to DNA samples and how well the assays work, and so on. That's already been done. There's no normalization that right now. I mean, we're going with what the software is doing already. So you can't play with that normalization that I know of. I really like these sample controls. So what this is a perfect time to now take those samples and say, I want to see those controls, and I'm going to configure those marks, and I'm going to highlight these. So I right-click on those samples, and I'm going to add this, and I'm going to call this CM63 controls. I'll type better. And I'm going to highlight those as, actually gray is not bad. You pick your favorite color, you hit OK. And then now, so those are always going to be gray now, no matter what marker you put them in. See, they don't always cluster perfectly, but you have to find a marker that they're actually separate in. But basically, so those samples, so this is a good use of the configure mark into different colors. I want to sample these controls as I go through. Okay, so I'm running out of time. So the two things we need to, there's 2 things we need to do to get the tetraploid data, which is what you guys are here for. So make sure you're on the full tab table. We're going to choose columns, and we're going to keep the index, the name, the address, delete everything else. So what I've done is I've clicked on chromosome position. I'm going to slide down, hit the shift key, highlight everything, hide the rest. And I also don't want, no, sorry, made a mistake. I'll undo that. I'm going to take away chromosome up to where the samples start, Atlantic Rep 1. Chromosomes up to fraction T, and I'm going to hide that. And I'm going to keep the samples, which is the rest. And I'm only going to capture the theta score, the data point. So I want to highlight genotype, score, and I'm hitting the control key, and R, and I'm going to hide those. Does everybody have that? Okay, so the only thing I'm keeping is theta. So I highlight all the others, and I hit hide. And hit OK. And now I basically have the SNPs, the address, and the data, the theta for each of the samples. And I'm going to export that. So I'm going to click the Export Displayed Data. So I'm going to call this SNP table, or whatever you like. So it's a text format. And I'm going to save it in my project directory. Hit save. SNP table text already. Do you want to replace it? I'm going to hit no. I'm going to call it SNP1. And it asks you, only selected rows columns will be exported. Would you prefer to export the entire table? Say yes. You want to view the file. If you hit yes, it basically comes up. It's not that interesting. It's a notepad. So that's all I need to export right now to capture the 4 genotypes, the 5 genotypes. What I'm going to do is actually I'm just going to minimize the program window, and I'm going to find that file. So if you go under My Documents, so I'll just highlight the folder, go into My Documents. I'm going to find the potato test data, and I'm going to open the 2 files. So I'm going to find my SNP table file. I'm going to right click, and I'm going to open that with Excel. Let's do it a different way. First, I'm going to open the Polyploid data converter. This is just a file that is actually specific to the 96 SNPs we have. So if everybody can open the Polyploid data converter. It takes a little time to open, but it will get there. This is a file that actually my technician or graduate student in my lab put together with a series of macros that can use the theta values to make the calls. So everybody should come up with this file over here. Basically, what Kevin in my lab, Kevin Stoffel in my lab has done, he's looked at the theta values and he said cluster 1 is going to be at 0.2 for this SNP, 0.55 will be cluster 2, and so on. He's captured the theta values that are in between each of the clusters for the files, is what he's done. And then he's got a series of macros which you don't need to know about. We're going to open now one more file. So we're going to go File, Open. And we're going to find our table, our SNP table. So we have to double click, look at all files. Where did it go? Under potato test data. And we're going to find SNP table 1. Okay? And we open that data. And delimited should be fine. Let's see, finish. Yeah, just delimited is fine. So you open this. What it does, it just opens your SNP table, and there should be 192 samples times 96 SNPs. And these are the theta values for all those. What we're going to do is we're going to capture the marker name and all the data. So everything from column B, actually you can even highlight column B, B1 to the rest of the data. Capture all that data. So I hit the shift key and I highlighted everything in between. Went to the bottom right corner, top left. Okay? We're going to copy that. So we're going to go copy. You can hit this little icon over here or hit Control C. That's in the clipboard. Then I can go back to this file over here, the Polyploid converter file. Has everybody got that? They've captured the data? We're going to click on marker name, and we're going to paste. So that's the data. And if everything worked, we're going to look at the genotypes. That's your genotypes. So there's a macro that captured the genotypes, and now you can, of course, you can zoom in to the genotypes. So what I've done, I've clicked to the genotype table, and there's a macro that captured that. So your samples in this case are only numbered in the same order that they are over here. But those are your genotypes with the 4 alleles that it was able to capture for this set of SNPs. Some of these guys, we weren't able to, the SNPs didn't work, so you can see some blanks. So the clusters were no good, or those SNPs didn't have good data. So they're blanks. So this Polyploid file converter is specific to the 96 SNPs only. So right now we've basically generated our own cluster files based on the theta values. And it seems to work pretty well, what we've done. I know Dave Douches has looked at it, looked at this data relative to others, and it seems to make sense. Well, what we're doing is we're working, the question is what kind of task is it to do for 8,000? So the task that I don't want to hear is to have my technician come up and do this one by one. We actually had a summer student do most of this, and we verified it. We had an intern do this. For 96, it was very doable. For 8,300, loci it won't be. Like I said at the beginning, Illumina, we're working with Illumina. We've provided Illumina with this data and the clustering, and they're actually developing the Polyploid data software so they can actually capture this data right from them. And you'll be able to export it right from the Genome Studio software. And so they hope to really have this in a couple months. So about the same time you should be getting your array data from the Infinium chip. We should be capturing that. So, question? So the question is, can you make this haplotypes instead of just genotypes? That's kind of the next step because we don't know how close these SNPs are. Most of these SNPs, some of these SNPs are, we know the haplotype already within genes, and so they'll really be connecting the data that Robin is putting together because she knows where these are within the sequence. And some of them really are haplotypes because there's more than one SNP per contig. And so that'll give you haplotype. But right now we're not there. But we've chosen the SNPs to try to, for most part, to try to represent as many genes as possible, except for the genes that, the candidate genes like that people are interested in where we may have chosen more than one SNP within those genes in general. So it's a lot of the haplotype part is actually defined on how you pick your SNPs, right, based on your sequence data. So that's in a sense before this. But when it comes to mapping data, if this was actually mapping data, then you can start defining haplotypes in the context of mapping because haplotypes are bins in a sense, genetic bins, which is what Chris is might talk about as well. Because if you don't have polymorphism between markers, even though they're unique SNPs in the genome, your mapping population may not have recombination between those two SNPs, and they become basically bins, which is equivalent to a haplotype in a sense, where all genotypes within that bin have the same haplotype. And so we're not at haplotypes there yet. We're not there yet, but that's really the goal is yes, look at haplotypes.
Begin to embark on QTL analysis at this stage with the beta values?
Question is, can you embark on QTL analysis with theta values?
They're different than standard QTL analysis.
I have, we haven't tried that. I mean, yeah, is that a relative thing? The end. Chris is going to answer that next time. Chris thinks yes.
Proprietary software. How do we as readers have access to the Genome Studio?
Well, the way we're set up right now is that SolCAP, at least with the SolCAP chip, so everybody's going to have access to a, you have to be able to access an Illumina lab, and it doesn't have to be our lab. It could be your own lab or a third party, and so on. So they're going to give you. They're going to export basically the files that you needed for Genome Studio or export genotypes, depending how close you are with that lab. So they can export genotypes. Right now, the only genotypes they're going to export are the diploid ones. So you'll be limited to that. You have to buy Genome Studio, unfortunately, to be able to play like we just did. But, so if you have access to a lab that has Genome Studio, you can do that. And so there'll be different levels of flexibility with your genotyping data where you can say, this is your genotyping data. We've called it based on what SolCAP clusters did it. Something you would ask your lab to do if you don't have Genome Studio. And it's fairly quick and easy. You saw how fast that happened. That's something you would have to work with your service provider, and we would certainly provide the files to do so. Is there a question from maybe in cyberspace?
No, not right now.
Okay. I'm sorry, can you repeat that? Does Genome Studio compute LD? No, it really doesn't. Genome Studio is really a software package to export genotypes. And from there you import it into mapping and QTL and LD and so on. So LD, we've used Structure, for example, is a nice Castle, and Structure are nice programs to basically upload this data into. And we've actually done that already with this very data set. It works quite well. Okay.
Alan, I think we're running late. We want to, I think, start the third session at around 4 o'clock. We're running a little behind here. Just to remind everybody, at the end of our workshop, there is a reminder that there's no free, nothing's ever free, and what we were asking you to do is to fill out a workshop survey. That's really important to us from our assessment of how things are going. The workshops are for you, and we want to make sure that the information that we're conveying is pertinent. So Michael Coe, who's standing right there, will be handing out the survey here to the people, and then the people online will be getting them by email. So let's take a couple-minute break, and then we'll come back for 4 o'clock.
Okay. Thank you.
Alan,